Morning, everybody. Spring's here. Welcome to the webinar. And I know Steve has a lot of fantastic information to talk about today. I'm excited.
Yeah, I feel like it's a little bit different webinar than some of the other ones that I've done. I guess more story driven. There's lots of little tidbits and weird stories and weird factoids. And sometimes I really, really love those. So hopefully everybody else enjoys. Thank you for joining us. Here we are. Almost into May. I can look out and I've got my hostas blooming and I've got tulips blooming. My apple is it's getting there. I've got some peach that are beautiful, smell great, and along with my plum. So thank you, everybody. Thank you for joining us. I hope that weather is good for you wherever you happen to be. And let's see, this month's webinar, I'm talking a bit about communication. And when I'm talking about communication, I'm talking about mostly the interactions that we have with plants and the microbiome, plants and microbes, and even some microbe to microbe. So the topics in general, the world around us, what's going on in the world around us, plant communication. I'll start on some stories about how plants communicate, and then microbe communication, and then that connection, that collaboration, that teamwork of plant and microbe. So question. What is communication? And I like this one. And that is why we lift on 3, because it's critical. It's important. If we don't communicate the right message, some pretty bad things can happen. And so by communication, what I basically mean is it's information processing. It's the sharing of information. It's an input of information, a processing of information, and then a reaction to that information. And there are all sorts of different ways that we interact with the world around us, and we have input from that world around us. So I like the example I have down at the bottom. That's a mantis shrimp's eye, which are just crazy, super weird, and super cool. So humans, our color vision, we see in we see in 3 different color spectrums. The mantis shrimp, its eyes are just weird. It has about 16 different colors, they think. And not only that, it can individually polarize different lights and different orientations. There's circular polarization, there's horizontal, there's vertical. It has eyes that are just bizarre. So looking at the same world around us, our eyes would see a completely different picture than what the mantis shrimp would see, because it has access to a wide variety of different visual cues that we don't. Another vision-based example is the way that our brain processes. Our eyes and brain kind of work, and for humans, around 60 hertz. So we see about 60 cycles per second. If you take a 100-mile-an-hour pitch, for human, it's a 100-mile-an-hour pitch. You take that same 100-mile-an-hour pitch and you throw it at a housefly, which it operates around 250 hertz, or roughly 4 times the speed. It appears everything is slowed down. So it's 4 times slower. So that 100-mile-an-hour pitch, only 25 miles an hour. And that is a pretty big difference. And that's why it's so hard. I'm getting some messages that I have no sound. Can anybody else hear me?
Yeah, Steve, Jenny's working on that. Sound is coming through perfectly clear here on my end. It might just be a volume issue with the user.
Okay, perfect.
So we're good.
All right, excellent. I'll keep going. So that's why it's so hard to hit those flies. The other end, the leatherback turtle, 15 hertz, so about 4 times slower. So that 100-mile-an-hour pitch feels like 400 miles an hour to that poor little turtle. So don't throw baseballs at turtles. I mean, come on. But what we're seeing is that our bodies, our brains, we have access to the information in different ways. Amy, my wife, hates this picture. I kind of like it because it's bizarre. But how do we access the world? And everything coming through an anthropocentric, human-centered worlds, we look at everything the way that we see it or hear it. So we have our 5 senses. We have taste, we have smell, we have sight, we have hearing, and we have touch. And that's how we process the information around us. I have a bunch more chats coming in, so I'm going to take a look again here real quick. Okay, looks like everything's okay. So if we look at our sense of smell, up in our sinus cavities, we have what are these, they look like little fingers that stick down. And if you zoom in, these are little olfactory ganglion. And the picture on the right kind of looks like a little hand. It's a funny looking little mitt to me. But basically those little hairs, those little threads that are sticking down, have little chemoreceptors, and that's the scent molecule. So you breathe in. Oh man, my sinuses sound clear. You breathe in through your nose, and then the various scent molecules that are in the air are going to interact with that olfactory ganglion, those little hairs. And that is received, and then a signal is sent to the brain. And that receiving is based on kind of a lock and key, a lot like we talk about enzymes, where we have different shapes that fit into those different areas, and it can make the smell completely different. And we have unbelievable specificity with our noses. So limonene, C10H16, same molecule, can have a pretty wide variety, a wide diversity of different odors depending on how that molecule is built. So these are enantiomers, they're chiral. Basically what that means, it's like left hand and right hand. Our hands are pretty much the same, but they're mirror images. And with this molecule, this limonene, if we have a D, or basically a right hand orientation of this molecule, then we have a lemon smell. If we have an L, which is again just like the left hand versus right hand, a mirror image, we change that smell completely. And I don't think I know too many people that would say that a lemon smells like a pine tree, but it's the same basic structure, and we can have differences. Again, depending on how that molecule is arranged, it can be a more orangey smell or a more lemony smell, but our noses are that specific to that specific shape. And it's totally a matter of opinion. Smells that I like and smells that I find appealing can be completely different. I mean, take the dung beetle, for example. If you've got, I don't want the same breakfast as a dung beetle. It just doesn't sound very good to me. But that's what their olfactory senses, that's what their sense of smell, that's what their communication, that information processing attracts them to. So the idea of terroir, the French, it's a loose translation for roughly earth flavors. So when we have these different microbes and different reactions that are happening in the soil, some of that smell, some of that flavor can make its way up into the wine or into the grapes or into the fruit, into our vegetables. Petrichor is a good example, and that's the general smell of rain. Most people, I love the smell of rain. Most people love that smell of rain. The smell of rain is associated with geosmins, and geosmins are mostly produced by actinomycetes and streptomyces. We grow quite a few different streptomyces groups, so if I'm ever in the mood for having a nice earthy, rich smell, I can just open up a bottle and I can get a good whiff of that earthy geosmin smell. Fruit flies, on the other hand, they do not like that earthy smell. That is an indication to them that there are either organisms, microbes that are growing that they don't want to interact with. Some of those can be mold, some of those can be bacteria, like the streptomyces, but they have an innate aversion to that geosmin smell. They don't want to lay their eggs there. They don't want to feed there. So again, the same molecule can have a wide variety of different interactions depending on the species. So what about plants and microbes? Do they communicate? Do they have access to the world around them? Well, yes, of course they do. They have to. Utilizing information from the outside world is critical to survival. One miscommunication key, kind of like that cartoon, you drop a patient, that's not so good. If you miss a communication key, that can be the difference between life and death. So kind of a fun thought experiment to kind of get you in the mindset of what I'm getting at is, what's your favorite place in nature? Imagine yourself there. For me, I love going to the beach. I love nice warm weather at the beach, nice salty water to splash around in, nice sand to play in. I also love going up to the mountain, going skiing, going snowboarding, playing in the snow. I love that. So now let's tweak it a little bit and imagine that you cannot move or speak. Now basically you're a plant there. Now all of a sudden all that salt water and that sand and that heat at the beach is no longer enjoyable. It's an obstacle to overcome. And same with all that snow, that cold weather that I as a human love playing in. Those trees, those plants have to struggle and fight through. It's an obstacle. So according to Dr. Ian Baldwin of Max Planck Institute, plants' genome has probably an order of magnitude more genes involved in environmental perception than most animals do. Most plants, they have to. They have to really tune their physiology and biochemistry to what is going on, and they need a very sophisticated system of perception and response. And that's absolutely true. I mean, having no ability to move yourself means that you are going to have to live through anything that comes at you if you can. And so we typically think about plants being stationary. They don't really move. And that's for the most part true. And these plants can also have a very complex sensory system. And in some cases, they can be very animal-like. But for the most part, even though it's animal-like, the sensory, and there are plants that have touch receptors, there are some of the fainting ferns, as you touch them, they'll fall away and droop away. And the Venus flytrap is a pretty incredible example. Not only can the Venus flytrap count, researchers have found that it has to have its touch receptors triggered about 3 times in about 20 seconds for it to be able to close. And if it doesn't receive that signal in that amount of time, it won't close. But the Venus flytrap is very, very fast. So I talked about those flies and how it's really hard for us to catch them because it's like everything is slowed down for them. But for a Venus flytrap, it's not an issue. So a fly takes off in about 300 milliseconds, 0.3 of a second. The Venus flytrap can close in 100 milliseconds. 1/10 of a second is how quick they can close when they get the right triggers. And the white mulberry catkin fires its seeds over 500 miles an hour. So generally, yes, plants are moving at a much slower rate than us, but there are examples in nature where they have a pretty incredibly fast reaction. And do plants smell? Do they have sniffers? And again, the answer is yes. So here on the left, those are those olfactory ganglion, those little fingers. And then on the right, we can see the plant cell transmembrane signaling mechanism. Same sort of idea, where we have this surface structure that is designed to receive a signal. And here's a fun little video. Hopefully the video will play. Penn State, Dr. Mesher was working with daughter, and it doesn't look like that is going to play. So let's see if I can get this. Not sure if this is working for you guys.
You got it. You got it playing, Steve.
Perfect.
In a series of experiments, they placed wheat and tomato seedlings in the same pot and planted a newly sprouted daughter vine between them. Then they set up a time-lapse camera to see if the seedling was actually making a choice. For hours, it circles the air like a snake, as if sniffing out its victims. And 9 times out of 10, its preferred victim is the juicy tomato, a tender plant that's easier to attach to.
You really get the sense of a behavior response. So really, there is some fairly strong selection here for this plant to make the right decision. Otherwise, you will die.
But how was the little stem making its choice? The team decided to play a hunch. They knew that all plants produce green leaf volatiles, chemical scents released by their leaves as they breathe. So maybe this predatory plant actually was sniffing out its victims. To test that theory, the team devised another experiment. First, they captured the scent of a tomato, essentially condensing the chemical odor released by the plant. Once it's distilled, they present the tomato perfume to the vine, along with a real tomato it can't possibly smell. Time after time, the daughter hones in on the chemical language that says, yes, I'm a tomato.
So that series, the What Plants Talk About, is incredible. It's a lot of fun, and it goes into a lot of examples of how these plants interact and communicate with them. So I've got the next video playing, so you'll have to give me a second. It was supposed to be an embedded video, so we'll get back to the fun here. So yeah, what we can see is that those daughter plants are receiving the smell, they're receiving those volatiles, receiving the chemicals, and it's communicating. It understands what's going on in the world around it by utilizing that sense of smell that it has. Okay, let's see. Another example, the smell of ore is what I call this one. Dr. Mesher, Penn State. And this has to do with goldenrod and the gall flies that like to lay their eggs and have their larvae grow inside, and they create those weird balls, the galls on the goldenrod. So when a male goldenrod gall fly is ready to find a lady friend, he will start producing a bunch of pheromones. They're attractant pheromones that will attract the female in. That's why I have the picture of Mr. Stinky down there, because he's putting out this smell, and that is to attract in the females. But the goldenrod kind of eavesdrops to those messages. It's able to also, through that same sort of perception, that same sort of sniffing, pick up on those odor molecules, those pheromones that the gall fly is using. And when it does that, it kind of kicks itself into gear. So the male is expecting to have a female come over and then have a great place for her to lay her eggs. But the reality is, what's going on in the background is the goldenrod has sensed those pheromones. And while it senses those, it increases its insecticidal chemicals, which decrease the female's ability to lay eggs. They also decrease the ability of the organisms, the baby, the larva of the gall fly, to consume the goldenrod. So it's this incredible interaction, it's incredible communication. And this is an eavesdrop communication. But again, it helps protect that plant. It wouldn't have been able to suppress that feeding as well if it hadn't been eavesdropping and listening. So it has this ability, these plants have the ability to control and shape their environment. An incredible example is wild tobacco. And this is Dr. Baldwin, Max Planck, who's researching those. I believe this is a patch of them in Utah, and different areas have slight variations to the different groups of the wild tobacco, but they are an unbelievably cool plant. So they're a pyrophytic. Their seeds require fire to germinate. Also, a lot of times you hear serotinous cones, same sort of thing that you see in some of the pine trees, but it requires fire to burn the resin off so that the seeds can germinate. And this one, they found that some of the organic chemicals, the carbon-based molecules associated with smoke and with fire, actually stimulate the seedlings to germinate. But anywho, so back to the wild tobacco. So it utilizes, since it's tobacco, it produces nicotine, which helps protect it from most insects, from most herbivores that want to take a nice chunk out of it. And when you have too much nicotine, bad things happen. Your muscles start to spasm, convulse, contract, and a lot of times your heart stops and you die. So that's a pretty bad outcome. Hornworms have figured out a way around that. They don't have to deal with that, and they can actually use the nicotine to help protect themselves. They'll accumulate it, and it can be used as an anti-predation molecule for them. So the hornworms are pretty happy to start eating in the wild tobacco, but as they start to chew on it, the hornworm saliva is communicated, it triggers a response in the tobacco, and it initiates a plant secondary metabolite production, an odor that attracts in the big-eyed bug, which is a predator of these hornworms. So the saliva triggers a response, and then these wild tobacco start spitting out a molecule that attracts in a predator for the hornworms. And if that isn't enough, I call a trichome treat an evil lollipop. So trichomes are generally a nice, sweet treat. The tobacco is purposefully doing this. It pumps a little bit of extra sugar, but it laces that sugary treat with a molecule that, once it's digested in the gut by the hornworm, it breaks itself down through digestion into a different molecule that makes the caterpillars stink. And in a stinky way that, again, tracks in more predators. In this case, it's birds and lizards. So now that the hornworms are being attacked by big-eyed bugs, they're being attacked and eaten by birds and lizards. And if the infestation continues, the plant will switch from nighttime flowering, which attracts more hawk moths, which lay the hornworm eggs, to a morning flowering and hummingbirds as its primary pollinator. Not only that, it'll change its flower shape, its opening time, and its nectar composition. So that original quote that plants have to be able to perceive and have to be able to adapt to their environment is 100% true in these wild tobacco. And it's incredible how plastic and how malleable they are. No time for a nap. So we've all seen around knapweed, very few plants are able to grow around knapweed. And that's because of the allelopathic chemicals that the knapweed is producing, the catechins that it spits out in the root, out through its roots, help shut down the growth and germination of other plants. However, there are a number of PGPRs, Pseudomonas butida, Bacillus subtilis, Bacillus ligniformis, as well as others that can degrade that catechin. So they can decrease the ability of the knapweed to send that signal out. Not only that, we see that lupine releases oxalic acid, which protects it, but not just it. It seems to help protect its neighbors. And again, as we have more neighbors that are able to grow, we have more carbon fixation, more sugar, more soil building, more microbes. The lupine itself gets more food out of it. So the debate is no longer whether plants can sense another's biochemical messages. They can, but about why and how they do it. The answer could have big implications. Farmers might be able to adapt this chatter to eating food, plants, or agricultural practices so that crops defend themselves better against herbivores. So utilizing the right species, the right blends of species, we can actually utilize some of this communication to help protect our crops. A fun example is in tomatoes, where these tomatoes, again, when they're under attack, they're spitting out a volatile. They're sending out a communication molecule. Hexenyl vicinicids, increase the cutworm mortality by 20%. So as the assaulted tomato on the left is being chewed on, it spits out this vicinacid, and then the tomato to the right of it picks up on that communication molecule, and now it's better able to protect and defend itself. Shocking discovery. Wounded leaves communicate their damage status to one another through a poorly understood process of long distance signaling. And we have a little bit better, hopefully this video will play. No, doesn't look like it'll play. I have these embedded and they're just not wanting to work right now. So in this video, what we see is researchers are utilizing this nerve-like signal. And what they found is, just like in human nerves, we're using glutamate, and that glutamate is causing a cascade of calcium to be spat out, and then that initiates the nerve conduction. So that can happen at hundreds of miles an hour throughout the body. Plants, again, they aren't that quick, but we see a very similar response where when the researchers would either damage the plants or they could just directly put glutamate on the plant, it would cause that signal to be sent around, and it would take about 20, 30 seconds for the signal to start moving, and then within another 20, 30 seconds, the entire plant had now received the signal that there is something going on. There's an attack occurring. And again, that's through glutamate and calcium. And it's that calcium channel that helps cue the plant and key the plant up to be able to protect itself better. Out of control. So another good example is when we accidentally do the wrong thing. A 2011 report found that commercial corn hybrids seem to have lost the wild maize plant's ability to release chemicals that attract parasitic wasps that kill stem borers. So again, before we understood what was going on, we'd been breeding different varieties of plants for a very long time. And geneticists, not knowing to look for some of these communication molecules, hadn't been keeping an eye out for them. And we'd actually bred out the ability of some of these hybrid corn to attract in those predatory wasps. So if we're utilizing refugia, if we're utilizing practices that allow those wasps to still be present, we have to make sure we match our corn varieties, and sometimes the older hybrids will still have that ability. Okay, so I've talked lots about plants, and plants are incredibly impressive. They're complex and large, but what if we go smaller? Can we have this conversation about bacteria? And bacteria are very, very small, so small that by themselves, they can't have an impact on larger organisms. But as we all know, anyone who's been sick or has had an infection knows this is not the case. And part of that is, again, through their communication. We'll talk a bit more about that. So can microbes sense? And again, the answer is yes, they definitely can. So bacteria are able to move, or at least the motile ones are, the ones that can move are known as motile. And a lot of times they'll have this bottom right, they'll have either flagella or cilia that they can twist around and spin around. It kind of works kind of sort of like a propeller that moves them through the environment. And what research has found is they're able to move in the direction of positive stimuli or away from harm. So if the environment is too hot or too salty or too full of other organisms, they're able to move away from that. Or if they find food, like when a plant sends out exudates, they're able to move in that direction, have ooh, piece of candy, because they're excited. It's a food source for them. So a fun example of that is Vibrio fischeri, and this is a bioluminescent organism that is found in ocean waters. So here we can see the bottom left picture is a bobtailed squid, and this is a little Hawaiian guy, and they're a few inches long, and they're a cute little guy or girl, and they are a nighttime feeder. The problem with feeding and being a predator at night is sometimes you will cast a shadow, and if there are any predators below you and they see that shadow, you become an easy target for them to eat. So the Vibrio fischeri, when in high concentrations, will start to glow. Vibrio fischeri are throughout the ocean. You find lots of them. The bobtail squid has created a symbiotic relationship with them by having what they call a light organ, and that's just basically a little sac or little sacs they have in their body that they encourage the Vibrio fischeri to grow in, and they feed them and they purposefully want to house them, because then when those Vibrio fischeri are in high enough concentration to start glowing, between the concentration of the glow and their ink sac, the bobtail squid can almost perfectly mimic the night sky above them. So now they no longer cast a shadow. They're able to have a light pattern that looks almost exactly like the moon and stars above them, protecting them from predators. But why do they glow? To glow or not to glow? Instead of a language, bacteria use signaling molecules which are released into the environment. And these are known as quorum sensing molecules. As you can see on the top right, these are intra-species communication. So between organisms of the same species, they have their own types of communication molecules. These are their own quorum sensing molecules. They're also interspecies, between different species communication molecules. So bacteria can sense who is around them, either that are the same species. They also are able to recognize who's around them that is not of their same species. So when there are enough of these organisms, they cause the Vibrio fischeri to glow by sensing the more of these organisms that are there, the more of this quorum sensing molecule that is spit out. And here you can see even the Vibrio fischeri molecule right there. That's what it looks like. The more of the organisms there are, the more that are spat out, the more that are spat out, the more that can be received. And once you hit a high enough level of quorum, then you're able to react. Rowinia carotivora is one that uses this to its advantage as well. And me being one who, you know, every once in a while you clean out in the fridge and you realize, oh no, I forgot about that. I've had some pretty slimy carrots and cucumbers in my day, and a few zucchini that are, you can't even tell they're zucchini anymore. So what this Erwinia is doing is it waits for sufficient numbers. And again, it's using these quorum sensing molecules. If there are a few of them, they're slowly spitting it out. And as more of them grow, there's a higher and higher concentration of them. If they attack too early, they know that they can be repelled by other organisms, they can be preyed upon, or the plant itself can help protect itself. But once there's a high enough concentration, and they sense that through their quorum sensing molecules, they, at the same time, all of them switch over. It's like a light switch. It's either on or it's off. They switch it on, and then they attack. All at the same time, they emit a virulence factor, which makes them more deadly. They spit out enzymes to create a wound or infection site, and they release protective molecules to keep all other pathogens away. So they are able to create that environment quickly and purposefully around them. And that is, again, through that communication. But the pathogens, that's not the whole picture. Don't think of all microbes. They're not guilty by association. Not one bad apple shouldn't spoil the bunch, because there are a lot of examples of very beneficial microbes. Here, Dr. Susan Simard, up in the Douglas fir forest in British Columbia, was doing research with ectomycorrhizal species. And what she found is sometimes trees that you would not expect to survive and be very healthy were doing better than they expected. And what the researchers found is some of the large old growth healthy trees, I call them mother trees in this example, are able to utilize some of their photosynthate, some of their sugar, and then they'll spit it down through their roots, and then through that mycorrhizal highway, that mycorrhizal network, they'll share some of that food through to other plants of the same species that aren't doing as well. And it's kind of like a nursery. They can help build and grow unhealthy or sickly or trees that would be shaded out otherwise. And so it can help maintain the health of the forest. And the way she did this was, it's pretty cool. She covered up branches with plastic bags, inserted some radioactive carbon-14, and then she could monitor and basically watch through the radioactivity. She could watch that radioactivity go throughout that tree that she infected with it, and then she could see it in the other trees as well. So it's being spread through the roots and through that mycorrhizal highway. And incredibly, we also see bacteria have what's known as altruism. Altruism is putting yourself before the team, and the little cartoon here is, tell me, Carl, are you familiar with the expression, take one for the team? And that little unhealthy gazelle is probably about to become a snack because he's just not going to be able to run away. But they're finding that different bacteria will do the same thing purposefully. An example has been found in E. coli, where certain individuals, a small percentage of the population, will purposefully slow down its reproduction rate to allow it to survive through an antibiotic. So if you have a lot of fast-growing, fast-reproducing organisms, they're going to take up that antibiotic very, very quickly, and will then kill them very quickly. So these, kind of sort of sacrificial, slow reproducers are still left behind because they weren't taking up as much of the antibiotic. So it allows them to survive through that antibiotic exposure. And so now, instead of the population dying, these populations are able to survive through what would otherwise have been a deadly encounter with an antibiotic. And scientists have found over 85 species that are able to do this altruism, slow down, and at one's own detriment, protect the whole community. And the list goes on. The litany of bacterial talents nibble at conventional assumptions about thinking. We usually think that microbes, the bacteria, are dumb. But bacteria can distinguish self from others, between their relatives and strangers. They can sense how big a space they're in. They can move as a unit. They can produce a wide variety of signaling compounds, including at least one human neurotransmitter. They can also engage in numerous mutually beneficial relationships with their host cells. And the list goes on. It's incredible what they can do. We shouldn't underestimate them. I love this example. And here we can see this is cordyceps that has attacked an ant. And here you can see it attacking a caterpillar, or not caterpillar, a tarantula, excuse me. So what cordyceps does is its spores will land on these ants or other insects, and then the mycelia, the hyphae, will grow in throughout the body, and they'll actually grow towards the brain. And they can actually hijack the brain. They can take control of the brain. And I don't mean like a little bit of control, like an unbelievable amount of control. And it makes me so happy that cordyceps don't come after humans. It requires, so for cordyceps to work, it requires a north-facing leaf of a plant about 25 centimeters off the ground, an environment with 94 to 95% humidity, and a location with temperature between 20 and 30 degrees Celsius. So this cordyceps that has hijacked the ant's brain has so much control over it that it will force that ant to climb, well, it's not even really an ant anymore, it's a zombie, to climb to that precise environment within 1% humidity. It's unbelievable. And the reason it does that is now it's in the perfect environment for it to shoot out all these weird, nasty tendrils that are full of spores that will then rain down and infect other individuals. Now, that is an incredible power to control. So we talked about plants, we talked about microbes. Now let's talk about the teamwork, that collaboration component where the organisms work together. And with these bacteria, they're small organisms, they can make a big difference. I've shown these pictures before. We can see an established fescue on the left, and that's the control. That's established. It's been growing for quite a while. Things are pretty good. Then after treatment with a blend of plant growth promoting rhizobacteria and biostimulants, 3 weeks later, we can see that major difference of what's going on with the roots. We can see those microbes working together to create a better home, creating that rhizosheath around the root, which is going to maximize nutrient uptake. So the plant is going to purposefully feed its friends because it knows that these microbes are going to be helping it. And feeding the friends is not just exudates of just sugar. We find that these microbes are using recruitment molecules. Again, they're communicating their wants and desires. So for example, the plant will spit out more malic acid when it's under attack. And I've talked about this before, but I'm talking about it now, associative communication, because this is purposeful communication. This is a dinner bell. This is a call. Remember, those bacteria through chemotaxis can move towards a food that they want. And in this case, the plant is purposefully spitting out the food that they know the bacteria want. So the researchers subjected this plant to Pseudomonas syringae, and then they watched as the plant spit out more malic acid and attracts in more Bacillus subtilis. And that Bacillus subtilis can then help protect the plant. Another example of this communication is with what's normally a powerful antibiotic, benzoxazinoids, which are produced by corn. But in this case, it's again using that chemotaxis to communicate a need and attract in Pseudomonas putida. So Pseudomonas putida is attracted towards those benzoxazinoids that help protect the plants, and they can actually feed on that. So these microbes can be incredibly powerful allies. Biofertilization accounts for approximately 65% of nitrogen supply to crops worldwide. Beneficial soil bacteria confer immunity against a wide range of foliar diseases by activating plant defenses, thereby reducing a plant's susceptibility to pathogen attack. So these PGPR, plant growth organic bacteria, have a wide variety of benefits that they confer onto the plant. They help protect the plant. If a plant's hungry, what can it do? Well, we talked about the RHIZOPHAGY cycle before, but when we take into account that the plant can purposely communicate a need, we now have a better understanding that through those exudates, it can attract in specific organisms to help feed itself. So the RHIZOPHAGY cycle, in a nutshell, if you haven't looked it up, it's very, very cool. Take a look, Dr. White's research. But basically, microbes are known to be great at mining nutrition out of the soil. Plants know this. So through their exudates, they feed them. The microbes are hungry. They eat a whole bunch of the exudates, that sugar, and they reproduce like crazy. They take nutrients out of the soil, and the plant sucks them in through its meristem, and then it partially digests them, squeezing out all that nutrient, and then it spits the microbes back out where they're hungry, starving, and right back ready to digest and fill themselves up with more food. So I think about these as like supplements. These microbes are little pills, they're little food for the plant. So if the plant's hungry, it's going to eat these microbes. The microbes are 10^2 plus traces, all that trace mineral that the plant can get out of these organisms. And depending on the different species that it's attracting in and the different species that are growing around it, it can have a different food profile that it's feeding on through that RHIZOPHAGY. What about if a plant is thirsty? Sure, the microbes can help with that. The PGPR produce hormones that help the plant control what's going on around it. So these soil signals, when a microbe senses a drought stress, it'll signal, again, through communication molecules to the plant that there's something dangerous going on down here. The microbes, we're sensing that we're running out of water, so you better be prepared. So what researchers have found, as with these PGPR in place, the plant is able to open and close its stomata more quickly and in response to drought conditions or abiotic stresses. And we can see a wide variety of interactions that are actually going on. So it's not just one communication molecule, it's a whole bunch, a whole raft of communication molecules. Now over here we can see VOCs, volatile organic. So these aren't even direct food, this is just the gas that the microbe breathes out. And by the plant interacting with that, we can see it decreases the sodium uptake, but it also increases sodium deposition from the shoot to the root. And then using acetoaminase, which we'll talk a little bit more about, and these different osmolyte molecules, antioxidants, cytokinin, all these different molecules the microbes are producing to help protect the plant. Not only that, if we have potassium mobilizing bacteria, but potassium is critical for stomata opening and closing. So when these bacteria recognize that the plant needs some extra protection from a drought stress, extra potassium can be mobilized, which allows the plant to open and close its stomata more easily. And this is a case of communication kind of being interrupted. And the microbes are doing that on purpose. Normally, when a plant is under stress, drought stress, it increases its ethylene levels. And that ethylene is designed to speed up the reproduction rate, to speed that plant along so that it can make it to reproduction, actually produce seed in that drought stress environment. But the microbes know that they can help protect the plant. So they slow down that communication of the plant with its ethylene. They actually decompose, they break down the ethylene. And with ACC deaminase, they're actually breaking down the ethylene, which slows that communication, which allows the plants to grow through that drought stress. Another more recently termed phrase is induced systemic tolerance. Plants inoculated with plant growth promoting rhizobacteria induce morphological and biochemical modifications, resulting in enhanced tolerance to abiotic stress, defined as induced systemic tolerance. So they help protect these plants indirectly from the drought stress as well, and it actually physically changes the plant's structure with that communication. What about if a plant is scared? So we know if it's hungry, it'll help, the microbes will help feed them. If it's thirsty, it'll help work through drought stress. What about if the plant is scared? Am I scared? I'm joking. But being attacked, if it has a bacteria, insect, virus, whatever's coming along, nematode, fungi, whatever's coming along to attack that plant, these microbes can help. And I mentioned this allelopathy, which seems like a little bit of a strange stretch, but what I'm mentioning here is, as we talked about with that knapweed, some of these microbes can actually shut down that communication. They can shut down that signal and digest that signal. They can modify those allelopathic chemicals, which will help protect plants around it, just like we saw with the lupine and the oxalic acid helping protect the plants around it. These microbes are doing the same. So when it comes to being attacked by a pathogen, pest, or whatever it is, there are two basic routes that happen. One of them is systemic acquired resistance, and that is like getting sick. The plant is attacked by a pathogen, it works its way through that attack, hopefully, and then it now has a way to defend itself. The other side is by using plant growth promoting rhizobacteria, which produce what's known as an induced systemic resistance. So this way, they don't have to actually be attacked by the pathogen. These microbes can warn the plant of what's going on around them, and through these communication molecules, the plant now knows what's going on in the soil environment or the phytosphere environment and knows what sort of threats are coming its way, and again, can help better protect itself without even having become sick. And I have down here, excuse me, note the current state of understanding. So we know the basics, and that by we, I mean scientists, human scientists. We have a decent understanding of what's going on, but we still don't fully know. We don't understand. We have unknown defensive compounds. We don't know fully what's going on yet. And that's kind of exciting, because that means there's still a lot of research and a lot of work ahead of us that helps us better understand how this induced systemic resistance and how this communication occurs. The term induced resistance is a generic term for the induced state of resistance in plants triggered by biological or chemical inducers, which protects non-exposed plant parts against future attack by pathogenic microbes and herbivore insects. It's like having a vaccine. And here we can see cucumber beetle. It's not just one type of pest, it's multiple types of pests. And here we can see Bacillus pumilus helping protect cucumbers from cucumber beetle. This is a soil microbe that communicates a stress, a threat that helps protect the plant. Bacterial wilt in cucumber, again, Bacillus pumilus in this case, helping protect the plant. Unfortunately, I don't think this video is going to work either. It's another little video, but I can describe it briefly. I can send around a link. But there are researchers were using Bacillus subtilis down in the soil, and then they were monitoring the activity of Pseudomonas syringae up on the surface of the plant. And without the Bacillus subtilis, the stomata of the plant was left open, and the Pseudomonas syringae are then able to go in like through a wide open door. Then they can easily infect the plant. Versus when the treated group with Bacillus subtilis in this case, when those plants were threatened with the Pseudomonas syringae, the plant was able to more quickly and adequately close its stomata and the Pseudomonas fungi could not get in through that open door. The bacteria on the root could help protect the leaves from attack, and I think that's just incredible. Callus. So I like to think about callus. It's kind of like a glove. So having these microbes down in the soil, again, communicates with the plant what's going on. And this induced systemic resistance can include, like I said, physical changes. So callus is a plant sugar. It's beta-glucan. And it is an enhanced deposition of callus-rich papillae is observed upon infection. So when the plant has exposure to these PGPR, it'll build up its callus. It's like building up a work glove, a protective glove around it. And so here we can see if there's a fungi that's trying to send a hyphae in, if it has a higher level of its callus, it gets trapped and gets stuck there. And in addition to forming a physical barrier for pathogen penetration, callus deposition are also considered to be a matrix for the accumulation of defensive compounds such as hydrogen peroxide, phenolics, various proteins, glycoproteins, and hydrolytic and antifungal properties. So having these microbes changing the structure, increasing the callus, and that callus also increases, it's like a sponge, and it's like a sponge filled with bleach. So now, if anything's trying to grow in there, it can't, because it has this protective environment around it. So what we see, what we think about, when we generally think about communication, nature's communication, our typical assumption is it's very, very simple, like this dashboard of this car. The reality is there's so much going on. We don't fully understand, but our takeaway is communication only works if we listen. We're just starting to understand what the language being spoken is, so we have more research ahead of us. But we need to respect the beauty and intricacy of nature because all life on Earth is extensively interconnected. So we've got to maintain diversity. Biodiversity is key to having this open line of communication. It's like a symphony under the ground and in the air, but we can only hear a couple of the instruments or a couple of the notes. We don't have a Rosetta Stone yet, but we need to recognize and understand as we're building these biological communities, as we're building this structure, this format, this forum, this matrix, this living soil system for the plant, around the plant, by increasing the living organisms, the beneficial organisms around it, we're working in ways that we don't even understand yet. We don't fully know what's going on. We understand some of the benefits, but we don't fully understand. I think that's very, very exciting. And I'm excited to talk again a few years from now when we have a better understanding of what communication is going on. And so with that, I hope that I shared some either fun or weird or some sort of fun example. So thank you, everybody. And I'm going to open it up for questions. Dennis, did you have anything you wanted to add?
No, excellent presentation, Steve. It just, I mean, it just goes back to what we talk about. You know, we so often think as adding biology is such a simple thing to build soil health or increase nutrient availability for the plant or root development, but it goes so much beyond that, based on not only the ways that they can help in agriculture, but also just the complexity of this system, not only with the biology, but also the communication with the plant.
Yeah. Oh, absolutely. No, it's incredible and it's exciting. And I didn't even go into Dr. White's research on virulence, where having these PGPR in the same environment as pathogenic fungi, these beneficial organisms, the bacillus and some of the others, will actually infect these pathogenic fungi, and they'll take away the virulence. They'll make them so that they're no longer a threat. They're no longer a pathogen. This, it's just so exciting because it's so unbelievably cool. But we kind of have to let these things happen. If we're trying to constantly control the environment, we have a pretty good chance of disrupting this communication, like we did with the corn. We didn't even know what we were doing, taking away that communication that the corn is supposed to have to call in the parasitic wasps. We didn't even know we were doing that, but we did it. So we have to kind of let nature run its course. Let's see. Should PGPR,
You got a couple of questions there in the Q&A, Steve.
Yeah, should PGPR products be applied only for grounds? Are they effective as a foliar mineral spray? So PGPR, plant growth promoting rhizobacteria, they're generally considered rhizo means root. So these are organisms that do best on the root. But like any of the things, whenever you say there's a rule, there's always an exception. So some of these organisms, they do just fine on the phyllosphere. So there's some Bacillus, Pseudomonas, there's some Streptomyces, there's some species that we generally associate with the root that can also do very, very well on the phyllosphere. And they'll grow and they'll create their own biofilms. And we talked a little bit about that in the foliar presentation last time, where we can utilize some of these organisms. So if we're using a little bit of a base blend of products, like a spectrum as a foliar, it will have organisms in there that do survive well in the phyllosphere. And they're also, that's just, the epiphytes, the ones that grow on the outside. The other side of that are the endophytes. There are endophytic organisms that will grow into the plant, and then we can have nitrogen fixation, we can have protection. So we can have these organisms growing throughout. So my advice is, think about these microbes like a coat of paint. You want to get that paint everywhere. You want to get those beneficial microbes protecting every part of the plant and around every part of the plant, because a lot of it is nutrient uptake. We're allowing the plant to have more access to nutrients, better access to nutrients. Do you want to add that, Dennis?
Nope, that pretty much sums it up with what we talked about last week or last month a little bit on the foliar application of using some of these beneficials to actually, especially when we start talking about critical points of influence or stress, it can be a great addition with our foliar applications. Oh,
definitely. And we have seen some little bits of research using some of these drought stress organisms through a foliar. We know that the VOCs that are being produced can be picked up. So if we do have some of those, the more of those that we have around the plants, the better chance we're going to have of that plant picking up on that communication and having those drought stress PGPR help protect the plant. So having them all over can be beneficial. Same with nitrogen fixation. And we can see that some of the phosphate solubilizers as well, they're going to help access that phosphorus and make that phosphorus available. What other question? Am I missing another question?
Yeah, there's a couple more. I think Faith had one. There's three there actually. Talking a little bit, can you speak to the use of biostimulants and autoinducers contained within to enhance and promote native plant growth, promoting and protecting microbes? And Bruce used to talk a little bit about that.
Oh, definitely. So it's a it's a hard one, because if you go over to Europe where they have pretty firm definitions of what a biostimulant is, and they can talk about them freely. Here in the US, I don't think that we have full nomenclature. They haven't fully defined what biostimulant is. But if you break it down, a biostimulant is just anything that encourages the growth and life. So it stimulates biology, stimulates microbe growth. So there can be a wide variety of different things that are biostimulants. There's some great products on the market that different companies are producing. There are enzyme-based ones. There are digested food-based ones, so taking different plant matter, different plant materials, and digesting them partially, and extracting out some plant-starting metabolites can be these biostimulants. Sugars, carbons, all these different materials can help the microbes grow. There's also been a lot of research on the prebiotics, some of these materials that stimulate the microbes to grow. There's some research, a lot of research in the human microbiome associated with the gut microbes and prebiotics and how they can help these microbes grow. So like inulin, for example. I don't know if there's as much research into prebiotics, but I bet there will be soon. I think I mentioned nanodots on one of my presentations a while ago, and that's an interesting one where we're seeing improved photosynthetic efficiency as well as nitrogen fixation with some of these materials. So yeah, they are measurable, and biostimulants do create a wide variety of benefits to the plant. It gets really hard though when you have to use metabolomics, transcriptomics, where you're actually looking at the gene transcripts of what the plant is actually doing to see how efficiently they work. But the long and short is we can see the benefit through nutrient uptake. So if we're utilizing leaf extract analysis, we can see these microbes are benefiting the plant by increasing nutrient uptake. The autoinducers, the example there, so autoinducers are like with our Vibrio fischeri. Once there's that threshold level, boom, the switch gets clicked on. Without that switch being clicked on, they're functioning in a different way. And it's the same with the Erwinia. So if we can repress the Erwinia to low enough levels, or we have organisms that are going to break down some of that communication molecules, because some of the microbes will do that. They'll purposefully interfere with communication. It's like playing a loudspeaker so that you can't quite hear what's going on. Some of these microbes will do that as well. So they'll suppress some of the communication. So some of the microbes can help with that as well. So if you shut down the signal, sometimes the pathogens can't turn that switch on. Native plants to grow. Yeah, no, I mean, these PGPR, there are a wide variety of PGPR encouraging native plants to grow. I consider these PGPR for native plants the same as you would for a crop. If we can get them in the root zone and we can get them associated with the plant, they can help that plant grow, whether it's through nitrogen fixation, phosphate solubilization, potassium mobilization, drought stress, and the list goes on and on and on. So yes, if you have native plants that you're trying to encourage to grow, you can utilize the PGPR. The hardest part usually is trying to get them into that root environment. We've got to make sure we do a spray that's appropriate that gets them to the root. If we just spray them on the surface and walk away, they're not going to do the job as effectively as I feel like we'd want them to. You want to add to that, Dennis?
No, I actually agree 100% when we so often talk about diversity and the key to diversity with the diversity of that, those organisms within that soil environment. It is so critically important, but we have to get them to where they can actually do their function and create a relationship with that plant. And sometimes in native, that is a little tougher than it is in a agriculture type scenario.
When you can more easily have access to the root. Oh, I agree. I mean,
yeah,
you've got to get the tool in the right place for the tool to do its job.
Which is one of the questions here is effects also important as seed germinates. And I mean, you can talk a little bit about Dr. White's research of how important that really is. I mean, we've always expressed it, but now we have that research that actually supports it.
Yeah, and Dr. White was saying, sorry, which direction of Dr. White's research are you wanting me to go this time?
How
Dennis?
important it is at when the seed germinates.
Yeah. And I didn't have that in this presentation. I did in another presentation, but he did research with tomato seedlings. And I didn't go deeply into the rhizophagy cycle, and we can do that, or you can listen back to a different presentation because we definitely talked about it a bit more there. And we can go deeper in if that's a topic that people are really, really excited about. I'm excited about it. But what he did is he sterilized tomato seedlings, and then he allowed them to germinate. And the seedlings that did not have any of the PGPR, that were still sterile, they didn't grow, and they didn't grow normally. The root tips, the roots that were trying to come out of that seedling were a dark color, and that's because of the oxidative stress. So plants are supposed to, remember when I mentioned rhizophagy, the plant kind of digests the microbe? What it's doing is it's using a bunch of reactive oxygen species to bombard and blast those microbes, and it basically rips off part of its cell wall, cell membrane. It's like poking holes in the wall. And so the microbes are expected to be the recipients of that reactive oxygen species. If there's no microbes there to digest, it's like having a whole bunch of digestive enzymes that have nothing to do, so they can actually start damaging the plant's roots. So the tomato seedlings had a much darker appearance around the roots. And then they also noticed that there were no root hairs. Without these PGPR, without these beneficial microbes, I think it was Micrococcus luteus that he was using in that situation, you didn't see root hair formation. So without these microbes, the plant needs those microbes to be spat back out, and that's what forms the root hair. The microbe grows in through the meristem, is partially digested, and then it's spat out through root hairs. So the more root hairs you have, it's generally a strong indication that you have a lot of beneficial microbes the plant is relying on to digest and extract nutrients from the soil. Without those microbes, there were no root hairs. So I mean, literally, the morphology of the root was completely different. With those microbes, we did see normal root hair, we saw normal coloration, and we saw normal growth. The plants, the tomatoes, have to have those microbes in order to grow right. So we have to have them there.
Well, and that's why we talk about so much with growers of how important and stress the in-furrow application, soil to seed contact with the inoculums at the time of planting, because we just see it makes such a tremendous difference in overall root expression and overall plant health. And, trying to water it down, as we were talking about a little bit earlier, when we have a native type prairie that we're trying to re-inoculate, or we have an agriculture field that we're trying to run it through the pivot of the irrigation system. When that seed germinates, we've missed that opportunity in some cases to actually have that biology right where we need it.
Yeah. Well, and looking back through, I mean, depending on, I think that I saw a question that was, discussing a redox that came up in there. And redox is reduction oxidation. The redox state of different minerals, is critical to its function. Like a good example is iron. I can either have steel, which is a usable tool, or I can have rust. And the difference there is the oxidation state. How many electrons, how many oxygen are associated with that molecule. One of them is usable, the other is not. The same can be said for a lot of different cations that the plant needs to utilize. So manganese, you can have it in different forms, of 2 or 3. One of those is the reduced form, one is the oxidized form. The plant can only utilize one form. The same can be true for a lot of different elements. So just because we have that element in the environment doesn't mean it's necessarily in a usable form. So maximizing these beneficial microbes helps that redox environment, and it helps convert some of those non-utilizable, non-usable formats to a usable format. And a lot of times we'll see some of that that oxidized state occur in like Dr. Kremer and Dr. Huber research showed, in environments that we've used a lot of pesticides. Some of these pesticides can have a direct impact on both the chemistry in the soil as well as the organisms that make up that soil. So if we've killed off a lot of the native beneficials, a lot of the reducing organisms like pseudomonads, for example, in that environment with our agricultural practices, we need to come back in and re-inoculate. And if we don't, it's just going to take longer, and we usually don't have the time to wait. It's, in my mind, kind of like waiting for the native seed bank to completely fill in versus adding some seed. If I want a cover crop in an area, I'm going to put out seed. I'm going to put out a multi-species cover crop seed mix to get those out there, because I know it's going to be faster. I can wait, but waiting in agriculture usually means missed opportunity, lost yield, and missed potential.
And that kind of goes into one of the questions that is here: food versus microbes. Are we risking upsetting the established of the beneficial relationship? If we add appropriate food, can we just support the growth of those beneficial organisms? And I think you just kind of answered that a little bit based on agricultural practices, based on tilling, based on herbicide, fungicide, insecticide uses, based on a monoculture. We have no diversity.
Yeah.
Lack of crop rotation. In a lot of cases, we have to add that diversity into that soil environment. And I talk a lot about habitat and how important building habitat is, or soil structure, because what we find is the better the environment or habitat is to support those microbial communities, the greater the diversity we can build within that soil environment.
Oh, definitely. If we, kind of take the theme of communication, if I take a couple of those threads and connect them, the idea of pesticides causing harm to some of the organisms. So pseudomonads are well known to be strong reducing organisms. Remember how I said the benzoxazinoids in corn are strong attractants communication molecule for Pseudomonas pseudomosquitida and that example. So if we've utilized a lot of chemistry that has depleted our natural pseudomonad species, and the plant now not only needs the reduced form of the minerals, usually manganese, but it also no longer has access to them because it's sending out that benzoxazinoid call. But if the microbes aren't there to respond to the call goes unanswered. So yes, the natives are critical, and using biostimulants is going to build up their populations. But just like in a normal agricultural fields, we're not quite sure who all's there. So just like with a cover crop, I want to put in, if I'm missing legumes, I want nitrogen fixation, I have to add nitrogen fixers. I'm going to purposefully add those species. Yes, some of the natives are there, and yes, they're probably going to help. But if I want to speed this process along, I'm going to use a purposeful approach. I'm going to add some of these beneficials, and then let the plant, like I said, let nature decide through these communication molecules. We're going to let the plant decide who it needs when it needs it. So we've got to put them out there. The troops, the food, the organism, it has to be there for the plant to be able to utilize it. I mean, if I'm building a house and I don't have any copper to do plumbing, I can't do plumbing. I mean, it's just missing. I have to add some of that back in. It's kind of the way I think about that.
Yeah, and we see that all the time in agriculture, like legumes, basically your peas. We use rhizobium specifically for that scenario. And and it has been shown that we get better yields. We get better nitrogen fixation than we do without that. So, I mean, that's a really good example. This has been used for, I mean, a very long period of time. It's not something that's new. Just looking at now diversifying based off of the understanding that we have now with the research that's coming out there and things Bruce used to talk about 30 years ago, how important all of these organisms are within that soil environment. And there's a lot of times where food, just biostimulants, food for that biology can work as effective as an inoculation depending on environment. And that's what we have to look at.
Yeah, carbon-based fertility is your route to help maximize. I mean, microbes, well, except for the photosynthetic ones, which would be an entirely different presentation, like the purple non-sulfur.
Purple.
they can so they're nuts. They'll photosynthesize, they'll fix nitrogen. If it's anaerobic, they'll switch over. They are a Swiss Army knife of microbes. So they are able to fix their own carbon, but they're kind of the oddball. They're kind of again, if there's a rule, there's always an exception. But most of these microbes rely heavily on the sugar provided by photosynthesis. Photosynthesis is the base of pretty much all life on Earth. We have to have it. That exudate, that plant's photosynthesizing is the base of pretty much all life. Microbes need to have it. So the more carbon, soluble carbon we can have in that soil environment, the more likely we are to have those beneficials, at least have a chance to grow. And I saw a question kind of pop through on, do PGPR always work? Are they always beneficial? I mean, no. I've seen examples where someone uses them in an inappropriate way or mixes them with chemistry that they're not compatible with, and then you don't see a benefit. When we use them in an appropriate way, an appropriate time, with appropriate crop, and like you already mentioned, in furrow at time of application, getting these PGPR around the root and giving them food so they can grow and establish with the plant is the best way to maximize their benefit. But if we don't pay attention to that, and if we ignore some of those, you can have tie-up. It's like using too much mulch and seeing some losses. If we're not using them appropriately, you can see a bit of lag associated with that, just like you would with mulch, where the microbes have consumed some of this nutrition and it's not available to the plant yet. So we've got to maintain that digestive balance. We want to keep our oxygen rates high, so we need to have a soil that doesn't have crusting so that we can keep our gas exchange flowing. Because if we have a lot of microbes, a high population, and then we convert or we have too much rain, we've got a very active environment, and now we switch to anaerobic, then we can see some detriment, because anaerobic digestion is very, very different than aerobic digestion. So we can have examples where a healthy soil kind of turns on us. But again, most of that is management based. Do you have anything to add to that, Dennis?
No, I actually, you kind of talked a little bit about what I was going to do, CO2 respiration, oxygen, how important that is to maintain. And, you know, a lot of things that we do in agriculture go totally against what we're talking about right here when we talk about flood irrigation and some of the things that we do that we can actually cause, in my opinion, sometimes more of a negative effect than a beneficial effect. And there's not many crops out there that want to grow in an anaerobic scenario.
No, they're extremophiles that will, but in general, no. Anne mentioned endophytes withstand the reactive oxygen. Somewhat. So endophytes, they have a little bit different role, and they grow in different areas. So the rhizophagy cycle, we're talking just about that root area. Endophytes can grow throughout the entire plant. So as we understand it now, that rhizophagy is happening in that root environment, and we're not seeing a similar digestion occurring in different areas of the plant. I don't know. I'm kind of excited to see if research goes in that direction. I kind of wonder if a somewhat similar digestion is happening in different parts of the plant, especially parts of the plants that would be exposed to more microbes, like the phylosphere, where we talked a little bit about with foliars, you have those channels, and we have to have an aqueous donor to an aqueous receptor. So we have to have a liquid environment. If I have liquid and I have food and I have a path for microbes, there's a decent chance that microbes are going to grow in that environment, just like a wormhole. That casing along the edge of the worm, where the worm has digested the debris, it's spit it out with mucilage to help create that hole, that burrow. I kind of wonder if a similar occurrence is happening on the phylosphere, in those channels, in those openings where you have liquid, you have food, you have microbes. I kind of wonder if that's going on. But as far as endophytes, they're a little bit different. They grow in different parts of the plant. Some of them grow in the cell, like you can see with our vascular mycorrhizal fungi, where they actually grow into the cell. And some of them are kind of growing in between the cell, so kind of in the cytosol, in that channel between. So depending on the endophyte, I wouldn't expect them in general to be exposed to as much of the reactive oxygen species. But I do like that idea, and I'll have to look into it more because it sounds like a fun one. But yeah, I mean, a lot of these organisms, they are aerobic, so they have catalase, they have dismutase, they have the ability to survive these reactive oxygen. But the plant is bombarding. I mean, in this zone, it's war. It's not a little bit of it. It is an attack, and it's purposeful so that it can digest.
And it just goes, I mean, when we start to think about it, I mean, there's research out there, Steve, as you know, that already proves, I mean, we see it on the seed, we see it in the stem, on the root, in the
everywhere.
flower.
Yeah.
I mean, it's everywhere. I mean, as you talk about all the time, I mean, when we start to think about the human body, you know, I think you have some numbers. I don't have them off the top of my head, but you take away the microbial community from the human body, there's not much left.
Well, yeah, I mean, the microbes weigh about as much as our brain on our bodies. So we're 10 trillion cells. They're 100 trillion microbe cells. 23,000 genes associated with the human the human genome, excuse me, 1 million genes associated with the microbes on and around us. So the idea of the holobionts, the entire living system, is critical. And again, more research has gone into the human than the plant, but it'll catch up eventually. And the more we look into human, the more we realize, oh my goodness, these microbes are doing so much more than we thought. And I mentioned in one of these spots, microbes produce at least one neurotransmitter that we know of. They're directly communicating with our nervous system, with at least one that we know of. That is insane. That is unbelievable. And I can talk about fecal microbiota transplants, FMTs, and all that, but I think that's not necessarily, this isn't necessarily the time for it. What kinds of Ron what kind of endophytes are available for spraying onto crops? Most of the commercially available ones that you see right now are azospirillum. Most of them are nitrogen fixers. A lot of them fall in that category from what I've seen on the market. There's some pretty interesting ones that are out there. A lot of your azospirillums are. Some of your bacillus will, some of your actinomycetes will. Actinomycetes are bizarre. They're called actinomycetes because they grow, they're fungus-like. So they're really good at growing in branching networks, just much smaller. So I mean, imagine your mycorrhizal fungi community is just this even smaller scale. And that can be pretty incredible for solidifying or creating that those aggregates, even on a micro scale. All right, oops, I'm sliding through. Oh no, I got into, even weirder things. There we go.
Imagine that, Steve.
The things that I didn't get to in this presentation that we'll have to talk about next time. Shane says go through it at half speed. Sorry, Shane. I feel like I went pretty slow compared to my normal. My normal. Yeah, and that's GABA, definitely. Gamma hydroxybutyrate, and it's an incredibly powerful neurotransmitter. And that's not the only one. I mean, yeah, the direct impact of the vagus nerve, the gut to brain nerve, that direct connection. There are microbes that directly communicate from our gut to our brain to that vagus nerve. It is so cool, and I'm so excited to see similar things for plants. All right, I think we've probably taken up a whole lot of time. So thank you everybody so much. Did we get through? I think we got through pretty much everything. And if we didn't, yeah, please email through. If this was fun, let me know, because I did this a little bit different than a lot of the presentations I do. And if you have topics that you think would be fun to talk about, fire them over. And thank you so much, everybody. I had a lot of fun.
Yep. Thanks, Steve. Excellent presentation.
Oh, thank you,
Thoroughly
Dennis.
enjoyed it.
Good stuff. All right. Well, again, thank you, everybody. I really appreciate everybody joining. I had fun.
Thanks. I'll see you soon.